Hello, my dear students. Welcome back to biology classes. So, today we are starting a new unit. The unit is having two chapters under your textbook, Biotechnology, Principles and Processes. And the second chapter is Biotechnology, its Applications. So, today we are moving to another branch of biology, that is biotechnology. And you are very familiar that this year the branch of biology has a greater scope nowadays. Okay, you know there is even BTech in biotechnology available in present days. So, dear students, in your plus 2 syllabus, you are learning the basics of biotechnology. We have already discussed what is biotechnology. Okay, so anyway, you should know a better definition for biotechnology. So what is your idea about biotechnology? When the word comes to you, what ideas or what are the other words come to your mind? When I say biotechnology, what are the areas you can relate to with that? Tell me. Yeah. So, you can relate gene cloning, tissue culture, okay, rDNA, like so many modern words are there. Then, let me start with our first chapter in biotechnology, that is Biotechnology, Principles and Processes. This chapter gives you the preliminary idea, the basic concepts regarding biotechnology. Okay, before moving to the chapter, you should know what is meant by biotechnology. Okay, so whenever we use any techniques to get products from biology, I mean the organisms, we can call it as biotechnology. That means when we are applying any technique to derive benefit from any organism, any living organism or any part of the living organism, we can call it as biotechnology. Don't think that biotechnology is something which is related only to the gene cloning, the rDNA technology, the gene manipulation, GMOs. Not so. I'll just tell you that when your mom prepares curd from milk using curd as inoculum, if your mom prepares curd from milk using bacterium, okay, what is the bacterium there? Lactobacillus acidophilus. Using such microorganism, when your mom or anybody if is preparing curd, that is a sort of biotechnology. Okay, so biotechnology simply means we are deriving products from organisms by a technique. Okay, whereas the modern biotechnology has a lot of areas. Okay, likewise we discuss gene cloning, GMOs, okay, RNA, I mean silencing, like so many things you are going to learn. Okay, so let us discuss one by one, I mean the topics one by one in the chapter Biotechnology, Principles and Processes. First time you study biotechnology in your course, school curriculum. Okay, then come to a better definition of biotechnology is given by EFB, European Federation of Biotechnology. Okay, European Federation of Biotechnology, EFB, has defined biotechnology as an integration. It is an integration of natural science with organisms or parts of organisms or their cells or parts of cells or even molecular analogues of the cells. in order to derive benefits, products or services. Products or services. So what do you mean by this definition? So European Federation of Biotechnology, EFB, has defined biotechnology as an integration of natural science with organisms, living organisms as such, or the parts of organisms, some of the body parts of the organisms, then the cells of the organisms or the parts of their cells, okay, the cell organelles or any part of their cells, then the molecules of the cells, in order to obtain products or to get some services. That means if we are using any of this, okay, to get products and services useful to mankind, that is what is biotechnology is. Okay, it's a simple definition. EFB has defined biotechnology as the integration of natural science with organisms, parts of organisms, cells, parts of cells or molecules of the cells in order to derive beneficial products or services. Okay, did you get me? Just study the definition as a good definition for biotechnology. Simply biotechnology means utilization, okay, of living organisms or their products or parts in order to get beneficial products or services. Okay, then let us move to the chapter. See, the first part of the chapter, we discuss what are the principles of modern biotechnology. So leave all other things, simple biotechnology facts there. Okay, now we are moving to the chapter is focusing on modern biotechnology. So, okay, the principles of modern biotechnology. The modern biotechnology principles, it is mainly based on, the modern biotechnology mainly deals with two principles. It works on the two principles. One is genetic engineering. The first principle is genetic engineering. Okay. The second one is chemical engineering. The second principle is chemical engineering. So you are very familiar with the term genetic engineering. So the modern biotechnology only we discuss now. It has the two major principles, genetic engineering and chemical engineering. Then tell me what is meant by the genetic engineering. What idea you have about this genetic engineering? Engineering with genes. That means we are cutting and isolating genes and introducing to other organisms. Okay, gene transfer technology, gene cloning, manipulation with the gene. Okay, if we are cutting and isolating a beneficial gene from one organism and transferring it to another organism, that is what is genetic engineering is. Okay, so any manipulation with the gene we can call as genetic engineering. Are you okay? So the genetic engineering principle deals with engineering works with genes. So remember, the gene, a beneficial gene from an organism is cut, isolated, and it is introduced to another organism, and if we require, we can clone it and we can express it, and the branch is nowadays developed very well. Okay, we will discuss. Okay, then the other aspect, the second principle, it is what? Chemical engineering. So suppose we are producing number of organisms by genetic engineering, genetically engineered, genetically modified organisms. In order to get better products or benefits, we are growing number of genetically modified bacteria. Suppose that. Then if so, that in order to maintain or in order to grow that bacteria or in order to grow the genetically modified organisms, we should have a better ambience for their better growth. Okay, when we make a better ambience for genetically modified organisms, that comes under chemical engineering. Okay, developing better ambience for genetically engineered organisms. Okay, that is what is called chemical engineering is. If these two areas are very well done or very well planned and executed, there is 100% success in the biotechnology applications. Okay, so the two major principles of biotechnology include genetic engineering and chemical engineering. Okay, come to the genetic engineering. Okay, so genetic engineering, now you know it's a manipulation or anything engineering works with the gene. Okay, then if it's genetic engineering, what are the major steps? What it mainly include? The genetic engineering works mainly include what? See, suppose human gene insulin. Okay, I'm just giving as an example. Okay, in human DNA, you have studied human DNA in very detail in the last session. Then in the human DNA, there is a segment, a DNA segment. This is coding for the production of insulin. Okay, so this part of the DNA we can call as insulin gene. Gene for insulin. Okay, suppose from the human DNA, we have isolated, we have cut and isolated insulin gene. That means the genetic engineering works, the main criteria I am discussing or the major facts. Okay, so the insulin gene from this DNA is cut and it should be transferred to what? An organism where we can multiply it. Okay, our purpose is to produce more amount of insulin from coding more insulin gene in bacteria. Okay, by getting it very well expressed in bacteria. Suppose one single insulin gene, in order to introduce this bacterium, suppose this is Escherichia coli bacterium. Okay, the bacterium is having its own gene. Okay, the bacterium is having its own genome. Suppose this is our insulin gene. Okay, see, anyway, the first, see, here what happened is, in this bacterium, you know, there is extra chromosome called plasmid. Okay, so for us to introduce this insulin gene to this bacterium and to multiply it along with the bacterium, when the bacterium divides, this DNA also, our DNA also divides. I mean, our purpose is, our aim is to introduce this to this bacterium. We should incorporate it to the genome of the bacterium. Then only it is able to multiply along with the bacterium. Okay, then for that, see, we can directly insert this genome of the bacterium. Only condition is the bacterium should contain ori. I mean, the genome of the recipient cell should contain ori. You know what is ori? Origin of replication site. It should not be cut and removed. Okay, by the time of our manipulation, it should not be cut and removed. If this genome has the ori and if we insert the insulin gene here, this insulin gene can multiply along with the bacterial genome. When this bacteria divides to 2, this genome gets doubled, this DNA also gets doubled. Okay, that is the usual way, simple sense. That means in order to multiply a gene of interest, here which is the gene of interest, insulin gene. Okay, in order to multiply our gene of interest, I mean our desired gene, insulin gene, in a host cell, if we want to multiply it in a host cell, what we need to do? We have to incorporate this gene, desired gene, into the genome of the host cell. Very simple fact. Okay, along with its genome, only it can, our DNA can multiply. If we are simply depositing it here and there is no scope for it to double or multiply. Okay, so in order to multiply, if our aim is to multiply this, to get its expression, what we need to do? We have to insert this gene into the genome of the bacterial cell. Okay, the second criterion, that genome must contain oocyte. Due to our insertion, the oocyte should not be disturbed. Okay, so that region must contain the oocyte. For what reason? If oocyte is there, then only this cell can replicate. Okay, replication is controlled by this site. If oocyte is disturbed, suppose when we cut and insert this, suppose this oocyte is removed. Okay, unknowingly the oocyte is removed. There is no use for incorporation. It cannot replicate if there is no oocyte. The two conditions you keep in mind. I am moving very slow because these all ideas are new for you. Okay, see, one gene from our DNA is cut. How it is cut, how it is transferred, all things we discuss. Okay, when it is cut and it is transferred to the host cell bacterium, we should ensure that the bacterial cell genome must contain the oocyte. Then only our DNA can replicate there. Okay, then and it should be incorporated to the genome, nowhere else. Then another important fact, most of the genetic engineering experiments, we are using bacteria as the host. Okay, and very interestingly, bacteria has an additional genome. You have heard it, you have studied it. An extra chromosomal genome is present in the bacterium. Other than this genome, normal genome, every bacteria, most of the bacterial cells contain what? An extra chromosome. This is a new, useful information. Okay, this information became very useful in biotechnology. That is about the plasmid. Understand? Bacterial cell speciality is it contains an extra chromosome called plasmid. Then all of you listen carefully here. A person, a scientist, if he wants to multiply our genome in bacterial cell, it is not compulsory that it should be incorporated to its normal genome. It can be incorporated to its plasmid. Okay, so we are hereafter we are discussing that side. Okay, so understand, in order to multiply a gene of interest, here insulin gene, in this bacterium, simply what is enough? Incorporate it with the plasmid. That is enough. Okay, for that here there is another way we are doing. Normally there is another procedure we are doing here. Okay, that we will discuss one by one. Anyway, just understand, if we incorporate, not inside the cell we do this, we discuss it later. If we incorporate insulin gene to this particular plasmid, okay, what is the status of it now? See, the insulin gene we have isolated and we have introduced it to plasmid. Now that is known as this DNA. It's a DNA, circular DNA. Plasmid is what? Circular DNA. Now this circular DNA is having a piece, an alien piece, okay, which we have introduced. So this is known as recombinant. It has recombined. Plasmid recombined with insulin gene. Now this is known as a recombinant DNA. Okay, so we can say a plasmid, if is inserted with insulin gene, it is recombined with insulin gene or any of the DNA segments, then we can call it as a recombinant DNA. Okay, so the genetic engineering is also known as recombinant DNA technology, rDNA technology. Okay, another name of genetic engineering is rDNA technology. So listen, my dear students, just keep the idea and get the idea properly. A recombinant DNA is nothing but a DNA with a foreign segment of DNA. When two DNA segments are combined, that is known as recombinant DNA. So, genetic engineering mainly deals with creation of recombinant DNA and its manipulation, okay, its maintenance. So, we can call genetic engineering as rDNA technology, right, okay. Then, so just listen, we are coming to the main steps in rDNA technology. The first step is, now we have discussed the first step, the first step is creation of, production, creation of what? rDNA. Genetic engineering mainly deals with production of rDNA. The first step or the main step in rDNA technology is creation or the production of rDNA. How it is produced? By transferring our gene or plant gene or any other gene of interest we can call, to a plasmid, mostly to a plasmid or any other alien DNA. If two DNAs are combined, okay, if any desired DNA is introduced to some other genome, that is known as a rDNA. So, the creation of rDNA is the most important aspect of what? Genetic engineering. And the second major aspect is gene cloning. Okay. If we are creating rDNA, if we introduce that rDNA to the bacterial cell, when the bacterium divides, it can divide. That means it is getting cloned. And when bacterium divides and produce number of copies, that many copies of our gene is produced. Okay, this is what we call cloning. Another important aspect of genetic engineering is gene cloning. Okay, and the third important aspect is gene transfer or the further technologies. Okay, and then the gene products, we have to derive the gene products. Okay, there are great devices. This insulin gene can be expressed and insulin can be produced. Recombinant protein can be produced. The production of recombinant protein. What I simply mean is, okay, when we transfer human gene, just listen to me. You know, nowadays diabetes patients are more and more. We need more and more insulin production. Okay, and when we transfer a human insulin gene, no need to isolate it from our body. It is artificially made by combining its nucleotides. That DNA is made artificially. The insulin gene is made and it is grown. Then suppose insulin gene is made and it is grown in bacterium. If it is simply remaining as insulin gene, what is the use? No need of insulin gene for us. We need insulin. So, we need to express that gene. Okay, that all techniques come under the production of recombinant protein. Recombinant protein here is what? Insulin. In order to produce insulin, we should have better expression facilities, gene expression criteria facilities. Are all things we will discuss in the chapter. This is just an introduction only. Okay. So, today, since it's an introduction section, I am not moving to the all details today. Just read the first pages of your book. That is enough for today, and just one paragraph only we reached. Since it's an introduction section, we can only do it so. Okay. So, once again, listen to these points. I am just briefing or revising. The definition of biotechnology you have learnt, and the modern biotechnology mainly deals with genetic engineering and chemical engineering. And when we listen to the genetic engineering, it has the major techniques like the creation of rDNA technology, the rDNA production and the gene cloning, gene transfer like things. And one more thing we have discussed is, in order to transfer a gene to another organism, we should ensure that our gene should be incorporated to the genome of that organism. Okay, and also what we should keep in mind is that portion should contain oocyte. These are the major things we discussed today. Okay, only these things. And for the next class we discuss who has produced the first rDNA. It's very interesting to know that, is it not? Who has got these ideas first? How, I mean, incorporating the two genes, okay, developing a recombinant DNA, something great, is it not? So who has developed the first rDNA? That is, okay, today I will give the scientist's name and the next class we will discuss on that. Okay, these scientists are Stanley Cohen and Herbert Boyer. Okay, so students, who developed the first rDNA are Stanley Cohen and Herbert Boyer. Okay, in 1972. He developed first recombinant DNA. Okay, so we will discuss about in the next class. Thank you.